This is a production of Cornell University.
Thank you, Ruth, for the introduction. As mentioned, I'll be talking about cassava brown streak disease. And just to let you know that cassava feeds about 800 million people in the world, most of whom are in Africa. But yield potential has never been attained, largely due to viral diseases like cassava mosaic disease and cassava brown streak disease. So today, my area of concentration will be cassava brown streak disease, and I will refer to it as CBSD throughout the presentation. So I'll talk about CBSD viral titer, root necrosis, and image analysis using the Pheno apps, GWAS, and a sneak peek into the results. So cassava brown streak disease is a positive sense single-stranded RNA virus that belongs to the family Potviridae. And we have two species of this virus, the cassava brown streak virus and the Uganda cassava brown streak virus. Both viruses are spread by movement of systemically infected stem cuttings and are also vectored by whiteflies. So phytosanitary interventions have been used to mitigate the effects of this disease, but we believe that deployment of resistant material is the most sustainable option. To evaluate this disease, a 1 to 5 visual scoring method is used, and a score of 1 means there are no observable symptoms, while a score of 5 means these are the most symptoms, so the severity is high. This method is quick and suitable for field assessment. So many studies have been conducted to try and understand genetic resistance of CBSD. But I'll talk about the most recent study from 2018 by Ismail Kayondo, where they conducted genome-wide association mapping and genomics prediction for cassava brown streak disease. In this study, they found that at 3 months after planting, when assaying the foliar symptoms, significant peaks were detected on chromosome 11. And at 6 months after planting, significant peaks were detected on chromosome 4, 11, and 12. Now, zooming into chromosome 11, they discovered that this is actually a nucleotide binding leucerin-rich repeat, which plays a crucial role in disease resistance. But they also noted that expression of significant peaks on chromosome 11 across environments were not stable. The authors of this paper later concluded that CBSD is polygenic and unstable across environments. So to expand on these findings from this paper, my research aims to provide a comprehensive study to uncover the genetic architecture of CBSD by complementing the 1 to 5 scoring scale with improved phenotyping methods like viral titer and image analysis. So therefore, I will be standardizing the triple sandwich antibody ELISA to expand on its sensitivity for high throughput titer quantification. The questions that you'd have is, why don't we use gold standard methods like the qPCR? These methods are expensive, and many studies have shown that if ELISA is carried out under optimal conditions, it could produce results that are equivalent to those of the qPCR. Secondly, we hope to undertake image analysis to provide image analysis for root necrosis to provide quantitative measures of the necrotic lesions across the cassava root. We also hope to identify markers associated to CBSD through genome-wide association studies. And in the end, we hope to understand differential gene expression when it comes to CBSD using RNA-seq. So the triple antibody ELISA uses 3 antibodies, and this method is sensitive, specific, reproducible, and affordable. Now, it's important that we develop such methods, we standardize such methods like ELISA to enable us understand the genetic mechanism of CBSD. But it's also crucial that we start selecting for low viral titer in our breeding programs. So selecting for low viral titer means that we will reduce disease pressure, which in turn will slow CBSD epidemics, and to a smaller extent reduce the chances of viral evolution. Currently, we are working with the Psycho2 population of the Ugandan breeding program. We are working with about 450 clones. These are being evaluated in 2 environments. We hope to collect titre data at 3, 6, and 9 months after planting to ensure that we study about viral progression throughout the growing period. Then while at 12 months after planting, we will use the root to determine viral titer using qPCR and also FTA cards. So to summarize the ELISA methodology, for the leaves, we are using pooled leaves since we are working with at plot basis, not plant basis. And then we conduct ELISA assays on these leaves. And from this, we look at the data as semi-quantitative and quantitative analysis. Then we generate the titre. And at 12 months after planting, we take the root and then carry out qPCR to identify the viral species and also the titer. And this is also done with the FTA cards. So we'll still have viral titer for the root using two methods. Now, in the incident that the ELISA, we don't trust the results from an ELISA plate, we have stored extra samples as herbariums. So we could conduct simple random sampling on the leaves and make sure that we carry out qPCR on those leaves to confirm the viral titer. So the reason why we are robustly looking at viral titer is that studies have showed that the virus replicates and accumulates differently in the leaves and the roots. So if we have to understand the mechanism of CBSD viral titer, we need to look at both tissues carefully. So from viral titer, we will be looking at root necrosis using image analysis. We're using the Pheno apps in collaboration with Makerere University. Now, it's important to know that root necrosis is the most devastating symptom of CBSD. It affects the root, which is of economic importance to us. But surprisingly, it's the least studied symptom because it's only evaluated once in the growing season, that is at harvest. So we think that understanding root necrosis using image analysis will enable us classify necrotic patterns. It will provide also quantitative measures like the number, the size, and the location of necrotic lesions across the cassava root. We also hope to understand relationships between these quantitative image-based necrotic measurements, viral titer, and the 1 to 5 scoring scale. So at harvest, the harvested root is sliced and images are taken at the proximal, the middle, and the distal part of the root. So this is an example of images taken from one experiment in one location, and you can see there's a difference with necrotic expression when across the root. So sometimes necrosis happens as a large necrotic patch, sometimes there are very small necrotic patches. Sometimes it starts at the center, sometimes it starts at the edge, and other times it's just random. So we think that to explore necrosis, we will have to generate data about the percentage of necrosis across the root, the size of the lesions, the number of the lesions, and the distribution of the lesions. So just progress on both ELISA work and the root necrosis work is we've assayed both traits in two regions that have that vary in disease pressure, and we're currently processing the data. So the next objective will be to use all these new phenotyping methods to find genomic regions associated with CBSD by conducting genome-wide association studies. Now, I'll talk about, the next thing will be to talk about just the trait heritabilities from the recent experiments that we harvested. It's good news to note that most heritabilities range from moderate to high, which actually means that the variability that we are observing is due to genetic differences. But the exciting thing about this PowerPoint, this slide for me is the semi-quantitative ELISA assay heritabilities and the quantitative ELISA assay heritabilities. This is the first time we are studying this trait, and the heritabilities are good to work with. Now, when we look at the correlations of the CBSD traits generated by the 1 to 5 scale and the quantitative assay, you can see these ones in blue that range from moderate to high correlation, positive correlation, are generated by the 1 to 5 scoring scale. But when you look at the semi-quantitative titre and the quantitative titre, there's a zero correlation between these two and the traits that are assayed using the 1 to 5 scale. So now this confirms our need to broaden the phenotyping methods when it comes to CBSD and include viral titer as an independent trait. So I will end here, and I would like to acknowledge my committee that's headed by John Luke, Michelle Heck, and Keith Perry. I would also love to acknowledge John Luke and Max Serra's lab, DMZ in Germany, and the best part is the team in Namulonge, Uganda, who have helped carry out my work, headed by Dr. Robert. I now welcome questions.
I think there's a question from Mike Gore in the chat, if you can see that, or I can read it.
Please read it. I don't seem to find the chat.
Yeah, Mike says, is it possible to sequence the viruses from FTA cards to ID viral strains and study viral population diversity and evolution across cycles of genomic selection? This would help investigate how genomic selection in the plant population shapes the virus population.
The answer is yes, I think it's possible, and we will test it since this is the first time we're using the FTA cards.
Okay, it looks like we don't have any more questions.
Okay, just one last. If anybody has feedback, please send me an email and I'll answer you.
This has been a production of Cornell University, on the web at cornell.edu.